Yeah, so I have been a little bit under the weather in recent days, so I'm hoping that I won't be making a Theresa May today, but we have made sure to vet everyone to make sure that there are no pranksters here, so hopefully we should not have that kind of issue. So I work as a group leader at the Sanger Institute, and I have with me today a research scientist, Vlad, and a postdoc, Talula, and in a little while we'll also have Davis from the EBI who will be helping out with teaching this course. So as the title implies, we're going to be talking about computational analysis of single-cell RNA-seq, and I think basically one of the purposes of this course is that all of the material is available online, which means that if there's something that you feel we went through a little bit too quickly, you can go over the material at home at your leisure. You may need to basically you need to download various types of information from GitHub, where we basically have a Docker which has all the packages that we are using here in this course. So on your machines that you have right here, they're already pre-installed, so you can basically just cut and paste any command which is in the course into your RStudio session, and it will run. If you just do it from scratch at home, you may not have all the necessary packages and dependencies installed. So Vlad, are you going to say a few words about?
Yeah, so there is a Docker image. So if you don't want to install things at home, you can run our course by basically downloading the Docker image using this command. So I've done this on this machine. So when you download it, you will see the log, something like that. And then once it's ready, you can go to, in your web browser, to localhost at port 8787. I have it here open. And then you use credentials RStudio. And if you sign in, you will have all the chapters available, and all the packages will be installed as well. And you can basically run everything you see on the website from here, and also plotting the images. So this is, if you don't want to install a lot of packages at home, you can just install Docker and run it from Docker.
Right. What I should also say is that currently this lecture or course is being live broadcast on YouTube. So if you don't want to be shown on the video feed, you should basically avoid sort of coming in between the screen and about here where I'm standing. And you can, of course, go home and watch this in slow motion replay if you're interested. I think, yes. So we teach this course about every 6 months here at Cambridge, but there is also going to be early in the beginning of next year. This is going to be taught in Berlin in February. So this is obviously not so much of interest for you guys who are already here, but in case you know someone who's interested in doing this stuff and prefer to go to Germany instead of Cambridge, this is an opportunity. Right. And yeah, so we assume that you, to do this course, we assume that you have basic understanding of ordinary bulk RNA-seq analysis, as well as a basic knowledge of R and Linux and Unix. If you have any questions afterwards, you're welcome to contact us. Most of the technical stuff is handled by Vlad, but other more content-related questions you can ask any of us. So, right, so I assume that most of you are familiar with bulk RNA-seq, which was a technology that became popular sort of in the midway through last decade, and it basically almost replaced the microarray technology that had previously been used for doing expression analysis. And what it does, very simply, is that you take a whole pile of cells, you extract the RNA, and then you turn that RNA into cDNAs, and then you put them through a high-throughput sequencer, and you get basically a picture, a snapshot of the transcriptome in this population of cells. So this is great if you want to be looking at sort of large number of cells, for example, the average profile of a tissue. But of course, it doesn't work very well if you're looking at heterogeneous system, because you can't get an understanding of what is going on at the level of the individual cell in terms of the transcriptome. So now, about almost 10 years ago, in 2009, the first paper was published where they were able to improve the protocol to such an extent that they could look at the transcriptome from one individual cell. But it took a while, basically a lot of technology development, and about sort of, I would say, roughly 5 or 6 years later, when several new protocols and much lower sequencing costs have made this technology much more accessible, and it's become a sort of much more widespread use since. So, as I said, the main advantage is that you can now find out the level of transcription at individual cells. I'm sorry, not the level of transcription, the level of transcripts at individual cells. And since sequencing costs are relatively low compared to what they used to be, you can do this for a large number of cells at a reasonable budget, and now, which means that you can start profiling complex biological tissues. And this is very important for situations where you have a small number of cells, for example, early embryonic development, or if you have very complex tissues where you think that most cells are very different, for example, in the brain. There are multiple protocols available. We will mention some of them. We won't be going over them. There are also many commercial platforms these days that one can use. However, all of that is stuff that is not really essential to this course, and we're assuming that we will be basically taking as a starting point the output of these experiments, which is the list of reads that comes off the sequencing machine. So just to give you a sort of very basic overview of a typical experiment is that you take your sample, you dissociate the cells, you isolate them, you lyse them, you extract the RNA, you reverse transcribe the RNA, the cDNA is amplified, and then it is put through the sequencer, typically a short read sequencer. You get your sequencing reads, and then you need to start analyzing those reads. So these are the steps that we'll be discussing in this course. So I've broken down the analysis here using this the flow chart here. It's color coded. So basically, the yellow stuff is sort of the kind of basic processing of reads that you would do for any type of sequencing experiments. So what you do here is nothing which is special to single-cell RNA-seq. So you can use sort of general purpose tools for assessing the quality of your reads and making sure that all of that is appropriate. The orange boxes here is basically the kind of quality control steps, normalization and processing where you need to use methods that are specialized for single-cell RNA-seq. And finally, when you're done here, you have a sort of quality-controlled, high confidence profile of each of your cells from your experiment. And then in the blue steps are sort of what I would refer to as a sort of the biological analysis, where you can sort of try to ask the questions that will help you answer the biological questions or issues that you were faced with as you started working on this project. So we will mainly be focusing on these two parts in this course. So as single-cell RNA-seq has become very popular in recent years, there have been sort of several different types of frameworks and pipelines that have been developed for sort of helping you out with the types of steps that I was outlined about. Some of them are mentioned here. This course is not a sort of automated pipeline or framework. It's more we'd like to help you to understand what's going on in at least in principle at each of these steps, so you can help evaluate which one of these methods are the best one to use, or if you want to basically sort of cobble together something on your own using the different tools that we're presenting here. So I think the main difference, obviously, between single-cell RNA-seq and bulk RNA-seq is the fact that in one case you're profiling millions of cells, and in the other case you're profiling only a single cell. If you only have one cell, you obviously have a very small amount of RNA to start with, which means that it needs to be amplified, and the amplification typically introduces various types of biases, and in the computational analysis, we need to make sure that those biases are appropriately corrected for and accounted for, so that we don't we're not led astray and sort of draw incorrect conclusions based on experimental artifacts. So that's sort of what you should keep in mind as conceptually what is the most important challenge here. So a short word about experimental methods. This is one of our favorite figures, which was taken from a recent publication from Sarah Teichmann's lab, where they basically look at time here on the x-axis, and each dot here is a sort of introduction of a sort of significant experiment, and sometimes we label the new techniques when they were introduced. And as you can see here, we have a sort of power law in terms of the number of cells that people are sequencing as a function of time. So today it's fair to say that most studies will you can sort of with only sort of moderate-sized budget for your experiment, you can easily do 10,000 or 100,000 cells, and I'm pretty sure that in a few years, sort of doing 1 million cells will be routine. So that, of course, makes it very challenging for us as computationalists to be able to keep up with this development and develop tools and methods that allow you to process all of this data that comes through this. So here are a list and links to some of these methods that are out there. I don't think the list is comprehensive, but it does cover most of the most popularly used methods out there. So if you sort of look a little bit more closely at these methods, I think there are sort of two different ways or aspects that we can use to categorize them. Is in terms of the quantification and capture. So for quantification, there are two types of protocols. They are either full-length or tag-based. So full-length means that you have reasonably unbiased coverage across the whole read, which allows you to quantify the whole transcript level, which is obviously very useful if you want to be looking at splicing or different isoforms. On the other hand, the tag-based, then you only look at either the 5' or most commonly the 3' end of each RNA, which means that you have much more limited information, and if your reads are relatively short, so up to sort of say 200 base pairs, it means that you're typically unable to distinguish many different types of isoforms, and you won't be able to identify most splice junctions. In general, the big difference between these two types of protocols is that the tag-based ones tend to have much higher throughput. So usually when you use one of those protocols, you can look at many more different cells, which is often advantageous. The other aspect is capture, which is basically how cells are captured. And there we look at three main types of options: microwell, microfluidic, and droplet-based. So microwell based, yes.
Is the reason that 3' or more high throughput just because you have more reads to work with?
Uh,
Read a shorter amount, or is there actually like an intrinsic reason that more high throughput method?
So I think no, so this is independent of the number of reads. This is how typically the reason is because it comes down to cost. So the cost per cell will be lower with the the tag-based methods, which means that if you have a fixed number, if you have a fixed budget, then you can look at sort of more cells, and then the other aspect is related to the sequencing depth, which is that if you're only getting the 3' ends, then you will saturate much faster. So it's not meaningful to sequence as deeply as it would have been for a full-length protocol where you have much larger diversity. So it will take you longer to sequence until you reach saturation. So apologies, I should have repeated the question. Question was about whether or not it was an intrinsic difference between the protocols because of the why you have more cells in one type and the other. So the microwell based protocols, essentially what you do is that you take your cells and you put them in some sort of you wash them out over a sort of plate like this, where you have lots of small wells, and you make sure that the cells are diluted in such a way that you will sort of on average get 1 cell per well, and then what happens is that you can carry out the sort of the type of lysis reactions and library prep inside of each well. So these are typically, although seqWell would be, of course, be the exception, which is a relatively new protocol, a relatively low throughput. Usually, people use either fax or you can even do it by hand with a pipette or laser capture to isolate individual cells and have them placed in these wells. So this is an important advantage of this is that if you can then also look at you can use a microscope to look down each well and take a picture, and that will allow you to sort of troubleshoot and sort of you can use various softwares to try to, or even look at it manually if you have a very small number of cells, to see if there are any issues in terms of, for example, the well being empty or there being 2 cells found in each well. So microfluidic platforms, for example, the Fluidigm C1 chip here, shown in this image, is a more integrated system for capturing cells. And essentially what you do is that you have the cells go through sort of, they're being pushed through these chambers here, and they're being sort of isolated inside the individual chambers, and you get and then you can the reactions take place, and you can sort of extract the material used for sequencing. The advantage of this is that it has higher throughput, but there are also some drawbacks. One of them is that you need to put through have roughly sort of 10 times more cells to push through, because not all of them will be captured. So you need more starting material. And another issue is that this is all based, this filtering is based on cell size. So it sort of requires that your cells are of roughly uniform size in your sample, because if it doesn't, then you'll have obviously have a much higher doublet rate for the small cells. So that can be more problematic. What has become very popular in recent years are droplet-based methods, which was the first 2 papers were called Drop-seq, where this figure is taken from, or in-drop. And the idea here is that you isolate, you use beads which are encapsulated in droplets, and then your cells are basically merged with the beads and the droplets. And then, so inside your beads, you have these tags and once the cell is inside the droplet, it is lysed, and then all of the transcripts will get the tags that were found inside each bead, and then you can basically have a unique addressing label to all of the transcripts that were found in that cell. So that gives you a very high throughput for your sequencing. Right. Any questions so far? So of course now the question is which of these platforms, or the ones that we haven't covered in this very brief overview, should I be using for my experiment? The answer is that it really depends on what your sort of biological question is. So there is no easy answer, and it also depends on your budget, of course. So what you really need to do is to sit down and think hard about what type of data you will get out of the experiment, and how that will compare to the type, how whether or not that is appropriate for the type of experiment that type of questions that you're asking. There have been, of course, a number of studies where these different protocols have been compared. So they take sort of the same types of cells, and then they churn them through these different protocols, and you can sort of see certain important differences. So one important difference that has been reported is just the number of genes detected. So here we're looking at 5 different protocols, and I think the most important thing to focus on here is the Drop-seq, which gives you very high throughput, but only gives you the 3' tags, versus the SmartSeq2, which is the most popular full-length protocol. So as you can see here, you have the number of genes detected is roughly twice as much in the SmartSeq2 versus Drop-seq. And obviously here you could also start looking at different isoforms, which you really can't do here in a meaningful way. Of course, the drawback is that this would be sort of more expensive than this. Right. So here's another study, where a sort of much larger group and larger number of protocols is being compared. They used something called spike-ins, which is something we'll be discussing later. So these are synthetic RNAs which are introduced. They sort of have known concentrations, so that they allow you to sort of calibrate your experiment. So here you can, for example, look at the sensitivity, which is sort of the minimal number of transcripts that are required to be detected. And as you can see here, the different experiments vary quite dramatically in terms of how low levels of transcripts that they are able to detect. So these are types of important considerations when you decide on which protocol to use. Okay, so now we're going to start talking about the first part of how to analyze your data. And as I mentioned, we start from the point of where you have where you sort of have you get a list of FASTQ files with reads back from your sequencing machine. And the first step, the first goal, is to create an expression matrix, which we'll be talking about a lot and using a lot in this course. An expression matrix is, by convention, it's a gene by cell matrix. So each row represents a gene and each column represents a cell. And the number in each element of this matrix represents, if it's, for example, counts, it represents the number of reads that were found for that gene in that cell. But of course, we'll be talking about different ways of normalizing this, these data. so that it may not be counts, it may be sort of counts per millions or transcript per millions. So the first thing you need to do with your reads is to QC them. And again, here you can use sort of standard methods. For example, fast QC, which is the one that we tend to use. So it is straightforward to use from the command line with your BAM file. And what you need to do then is to sort of figure out if your reads are all just garbage or if they actually do something useful. So here is an example of output from the fast QC. So where we plot the position of the read and the quality scores, which is provided from the Illumina machine. So what you can see here is that we have a little bit of worse quality at the start of the read and at the end of the read, which is quite typical, what you'd expect. Somewhat surprising, we have a dip sort of somewhere here in the middle where we have a reduction in the quality scores. For this experiment, we don't really know why that happened. We think that this is only sort of a handful of bases in the middle where this appears to be a problem. So we hope that this should not cause too much problems for the alignment downstream. So we're just going to make a mental note of this and proceed. But if there were more errors here, you might want to sort of consider doing more careful checks, and potentially you'd have to rerun an experiment if things were really bad. Yes? Sorry? So I think it depends on your experiment, right? Do you want to answer that question? So the question was, do we have one file for each of our cells for the whole experiment, or different?
So it sort of depends on the sequencing facility you use. Some sequencing facilities will demultiplex your data for you before they send it to you. Sometimes they won't. And generally you'll have multiple cells per lane, but you might have, so for this experiment, I believe we had 4 lanes for 200 cells. You might have 4 lanes for 10,000 cells. You might have 1 lane for 100 cells. It depends on how deeply sequenced and how many cells you're looking at.
And if you do have a lot of cells, like 10, 20,000, are all these QC methods appropriate? Can they all take really big files?
They can all take really big files. They're designed for huge genome sequencing as well as RNA-seq. So they're all mostly optimized. The only one I'll put a caveat is the mapping. Different mapping algorithms have different speeds. So the one we're going to talk about is fairly fast. And for 10,000 cells, it would probably take a day or so to run, which may or may not be fast enough for you. There are faster ones available as well.
Right, so once you've ensured that you have good quality of your reads and potentially thrown away the ones that have poor quality, what you need to do is align the reads to your reference genome. So here there are essentially 2 strategies that you can do. You can sort of do the more traditional way, which is sort of first you do the alignment using, for example, star or top hat or BWA or whatever is your favorite aligner, and then you go on and quantify it. Or if you have a very large data set, you may want to, you probably don't want to be using that strategy because it will take a long time. You want to be using pseudo-alignment methods, which is called, for example, Callisto or Salmon are the 2 most popular ones, where you instead of doing first alignment and then quantification, you do both of these in one step. So we're not going to be talking about that in a whole lot of detail, but we'll be focusing on the first approach here, where we first do alignment and then quantification. So here's an example of how to run star. So again, this is not the purpose of this course. It's fairly straightforward doing this from the command line. What's important to keep in mind is that if you have spike-ins, you need to add those sequences to your reference genome to make sure that they get quantified appropriately. And you also need to make sure to process your UMI barcodes or cellular barcodes before you do the alignment. So an important question is, of course, so what can I expect in terms of alignment rates? How many of my reads are good? The answer to that question is that it really depends on your organism and on your protocol and so forth and so on and so forth. So there are no really hard rules here. In our experience from the data sets that we have been looking at, which are primarily from mouse or human cells, we typically get alignment ratios or mappable ratios of around 60 to 70%. I think the important thing to keep in mind is that whatever you have, you should expect it to be sort of roughly a normal distribution around sort of the mean value across all of your cells. So if you do find a cell which has exceptionally poor or exceptionally high alignment rates, you should be suspicious of that and quite possibly exclude that cell as an outlier from further analysis. Right. So next step is to do so here we're talking briefly about how to do the sort of mapping and quantification using Salmon. Again, it's very straightforward using as a command line tool. So if we do the traditional alignment, we can do some further QCing here. So what we've done here is that we've plotted, and so here each bar corresponds to a cell, and the height of the bars correspond to the number of reads for each cell. And then we've colored them depending on what fraction, the number of reads that we're uniquely mapping, multi-mapping, and unmapped. And then they're sorted. So what you can see here is that we have 3 cells here in the beginning where you have a very sort of low number of uniquely mapped reads. So we have fewer than 1 million uniquely mapped reads. So we will consider those poor cells as very likely that we didn't have an intact cell to start with there. So we're going to exclude those from the further analysis. Other than that, everything looks pretty much what we'd expect, except sort of here where you see that you have, in general, you have a sort of steady increase of both the number of reads and the number of uniquely mapped reads. Except here you see you have 2 cells where you suddenly have a drop in the number of uniquely mapped reads. We have no idea why this is. We decided to keep these 2 cells because the number of reads, uniquely mapped reads, is still a very healthy number. What I would recommend is what you should do is probably make a note of these 2 cells, which they were, and for your future QCing, keep an extra eye on them to see if there's something weird going on here. But in this example, we didn't find anything odd about them anywhere further downstream, so they were used just as normal. Right. So to further QC the mapping, we can consider the coverage of the genes, which is information that we're really interested in. So we can, for example, use the tool called RCQC, which will output this type of meta plot, where I plotted here on the x-axis is the the relative position in a gene. So 0 would be the 5' end, and 100 would be the 3' end. And here on the y-axis, you have the coverage. So this is supposed to be a full-length coverage transcript. So the first thing you can notice, it's not truly unbiased coverage, but this is pretty much par for the course, what you'd expect. So what you can see here is that you have Each line here represents a cell. This is actually the same data that we had in the previous plots here. So it's 90 cells or something. So most of them behave in a similar manner, but we do have 3 outliers here, where you, for some reason, have significantly lower coverage in most of the gene body and have almost all of the reads coming close to the 3' end. So in this case, Tallulah, did we exclude these cells or no?
Yes.
We excluded them, yes, because they are outliers. Yes, question? Yes. So, yeah. So the question here is this the reason why we have differences here? Is that because of cell size differences, or is it because of amplification biases? I think in this case, do we know the answer to that?
In this case, we believe it is amplification noise because these particular cells we expect to all be the same size approximately, but cell size will also cause a bigger skew usually than is seen here. And I would point out this distribution also depends on the method. So droplet-based methods tend to have a much more extremely skewed distribution, whereas this was SmartSeq 2. So this is what SmartSeq 2 distribution looks like.
So the question is if people use ribosomal depletion, and I think the answer to that is yes. No.
Almost all of the methods use poly-A selection.
Oh,
So
okay.
that will
So
remove.
So there is selection, but it's not depletion, but it's through poly-A. No, so the 60 to 70%, is that coding, or is it the entire genome? So that's for the entire genome. But most of that will be coding. Right. So finally, once we have excluded the cells that we thought were of poor quality, we can quantify the reads. So here you need to use another method, for example, HT-seq or feature counts. There are several of them available. And you also need to provide an annotation for your genome so that it knows which genes method knows which genes that you're interested in. And then you'll get an output which includes the number of reads that were found in each of those genes for each of the cells that we're using. And of course, if you are using the pseudo-aligners, this step is not necessary. So now we're going to talk about unique molecular identifiers, or UMIs for short. So as I mentioned, one of the challenges in analyzing this data is that you have an amplification step. So where you take your cDNA and you put it through a number of steps of Pcr amplification before you sequence it. It's well known that the Pcr amplification is not unbiased, and it can, in particular, there's a strong GC bias going on here. And what happens is that transcripts that have either very high GC content or very low GC content are not being as highly amplified as the ones that have intermediate content. So one way of trying to combat this problem is to use UMIs. So the idea behind UMIs is that you have a short sequence, typically no more than 10 base pairs long, that is being attached to each transcript prior to amplification. So then, after you amplify, after you sequenced, if you find two sequences that appear to come from the same transcript, and then have different UMI tags at the end, then you know, you would assume that they were the cause. They came from different molecules before amplification. Whereas if they sort of map to the same location in the genome and have the same UMI tag, they're most likely to be caused by the same sort of come from the same original template, and hence they would be considered sort of duplicates. So this basically allows you to figure out what was going on before the amplification step, and it should, at least in theory, give you sort of much more accurate quantification than if you're just using the reads, where you don't really know what kind of biases you got. So when people design their experiments, this is sort of a typical schematic where you'll have a transcript, you will have a poly A tail, and then what you'll do is that you'll add your UMI, and then what is often done, especially in the droplet-based protocols, is that you also have a cell barcode. So that's an additional barcode which allows you to figure out not only which molecule it was, but also which cell it came from. So what you need to do in terms of the processing is that you need to keep track of all of this information. So before you start doing all of the mapping that we were talking about before, you need to look at how the experiment was designed. You need to first sort of trim your cell barcode and sort of split all of the reads into sort of different buckets, depending on which cell they came from, which you can identify by this barcode. And then once you're inside the cells, you can in each individual cells, you can look at the different UMIs, and you can identify unique molecules. Yes. So I think it depends on how you design it. How long are the barcodes and how long are the UMIs? And I mean, the cost of having a long UMI and a long barcode is that sort of you have less information about the transcript. But the advantage is that you'll have a greater diversity. So it really depends on the sort of the number of cells that you have, and the number. And here, the cell barcode is a function of the number of cells, and the UMI will be a sort of somewhat a function of the sequencing depth that you're using. So how long are the reads? It depends on your technology. I think typically most, at least the Sanger facility, they offer sort of different read lengths, so you can choose what you want. But I think you should expect sort of 125. Is that fairly standard these days? And you can also have either single read, single end or paired end reads. So the problem with UMIs is, of course, they're not perfect. So you can have different types of errors. One error you can have is that you can have an error in the UMI. So what that means is that you can have two reads that originated from the same molecules, but because of errors in the UMIs, you think that they end up from different molecules. So different estimates suggest that these errors are on the order of 5 to 10%. So it can be significant. So this is something that you do need to consider. You can also have errors in your reads. So it means that even if you had something with two reads that were identical and have the same UMI, they end up being mapped to different parts of the genome, and hence they end up being double counted that way. And you can also have collisions. So if your UMIs are too short, you can have two reads. If you have a very diverse transcript, you can have two unique transcripts that end up with the same UMI, and but then you think that they came from when you look at the data later, you think that they came from the same molecule, even though they didn't. So there are different ways of trying to estimate these types of issues. And there are several different tools that are being developed. One of them is called UMI tools, which specifically tries to deal with this issue of errors in the UMIs, and really the strategy here is what typically tends to happen is that you will have most of your most of the reads will sort of have a single will have a specific UMI, and the errors will be sort of much less frequent, and you can use that to kind of filter them out. And then you can also, and for the collision probability, the problem of having sort of multiple molecules from the same transcript or the same origin in the genome having the same UMI, you can calculate this in a sort of probabilistic manner. So this is something we will look into very shortly. So here's some data to illustrate the issue. So here we have an experiment where we plotted the number of UMIs on a log scale versus the number of reads. So ideally, we would hope this to be a sort of a straight line here. And we see something which is fairly close to that. So this suggests that, after all, they do seem to work quite well in terms of the quantification. As we saw before, we have a sort of a big difference in terms of coverage, in terms of the number of reads that we get from each cell. Now, this is, of course, also sort of reflected in the UMI analysis. So here we've plotted from an experiment. We have each dot here is a cell, and we look at the total number of molecules that we found for each cells versus the total number of genes that were profiled in each of those cells. So we see that we have a sort of large diversity here again. This is what we label the capture efficiency. So this leads us on to your first exercise. So now you need to sort of go to your RStudio. You need to load two data sets, which corresponds to the UMI counts and the read counts from an experiment. And then you need to use this data to try to carry out these two exercises. So I will leave you to work on that for a few minutes, and then I will be handing over to Davis, who will be talking about bioconductor and skater.